So today is the 6th October 2020, and it's quite special that we have Clayton Deighan from NanoView Biosciences, actually our first industrial speaker here. That's going to talk about heterogeneous tetraspanin display on EVs, big, small, and various origin. So Clayton has been studying methods for detecting EVs and working with scientists around the world on their characterization for the past 8 years. Most recently, he works for NanoView Biosciences, which developed tools for single vesicle characterization. So without further ado, please welcome Clayton.
Thank you, everyone. Well, we'll get started. This is a little different for me coming from industry. I know a lot of people on the call have probably met me in the past working with Malvern and NTA. I got my own start in EV analysis in grad school at Ohio State back in probably 2012, 2013. Used NTA a lot and a lot of other common techniques. And then I was a field application scientist for Malvern Panalytical for 4 or 5 years. And right now I've been with NanoView Biosciences working on SP iris and fluorescent detection of EVs for about the past 2. So I'm sure many of you have seen my sales presentation, seen me come in and give a presentation about the technology. Today's talk, we're going to focus a lot more on the data. I'm going to be very, very brief with the underlying, how does it work, what does it do? And we'll approach it from the angle of, if we can detect individual EVs and we can analyze their biomarker content, what does that help us learn about the display of even common biomarkers like the tetraspanin family on EVs? And that's where I want to keep it today. So ask questions, get involved, see what you think, and we'll take it from there. A little overview. We'll do a quick motivation, brief, as I said, technology review. It's not the focus of this. We'll start at the fundamentals of tetraspanin colocalization, and we'll start by challenging some of the assumptions about the difference between surface budded and endosomally formed exosomes or extracellular vesicles or microvesicles. Then we'll take a little dive into the tetraspanin display on distinct but similar samples where we look at cancer cell lines versus plasma-derived EVs. So one's formed in the body by the milieu of cells that contribute to plasma EVs. One's formed in a dish by cancer cells. They have surprisingly similar displays of the tetraspanins, but very different loading of different markers. And then we'll look at utilizing the colocalization aspects in complex mixtures to track the subpopulation within the plasma. And I think that'll leave us at a pretty exciting place. So I think the underlying motivation for this talk is, it really harkens back to that same question I think a lot of us are still asking, is what is an extracellular vesicle at its core? And I think by nomenclature today, there's still 2 primary subsets that we think of. We think of exosomes, and those are which form in intraluminal vesicles called multivesicular bodies. And I'm not here to be a biologist or blow you away with any of this, but multivesicular body combines with the cell surface and releases the nanoparticles into the extracellular space. And we'll contrast those to what are commonly termed microvesicles. And my personal preference is to lump these all together and call them extracellular vesicles at the moment, but microvesicles being those which bud directly from the plasma membrane through any number of pathways which are known to be available. These are often more numerous than those of endosomal origin, and we'll see how even with separations, it's quite difficult to pull apart these 2 populations into distinct differentiable groups where we can say with certainty, these come from the surface, these come from the inside. And in today's talk, we'll explore the display of the common tetraspanin markers in these different populations, and we'll examine some assumptions about the size difference or composition that may or may not hold true when we take a deeper look. So right off the bat, we've got our microvesicles budding from the surface. We've got our exosomes coming from multivesicular bodies. Whether we're looking in cell culture, where these would be together, or whether we're looking in a biofluid, where again, these would be in a mixture, it doesn't really matter. We want to know, can we easily separate these 2 populations by physical forces anyway? Starting at ultracentrifugation, obviously the most common, in my opinion, on dead-end ultracentrifugation. We all know sequential ultracentrifugation techniques, the 20K spin followed by the 100K spin. Do those actually pellet, particularly microvesicles in one or in the other? With the dead end, not really. You're going to pellet some fraction of what's in the tube, and it's going to go to the bottom, and then you're going to take off the supernatant, and then you're going to pellet again, and you'll get a different pellet. But the overlap between those samples is certainly possible because they're both going to move to the bottom of the tube at different speeds. If we have a density gradient and we suppose that the microvesicle or the exosome could be different in its density, then we could certainly separate them by floating them on a gradient. But that's not well demonstrated that microvesicles actually separate from exosomes, although vesicles may separate from other contaminating nanoparticles, particularly lipoproteins or chylomicrons, which we'll visit in a few slides. Size exclusion chromatography, same issue. There's no separation that's going to drive apart the microvesicles from the exosomes, because when we do these SEC for vesicle isolation, we're often focused more there on removing the protein fraction. And with the design of most of these columns on the market, we're actually voiding out the vesicles. We're not separating them on size by having them probe the packing. We void them out, and then we let the protein probe the packing and elute in a later fraction. So we collect the first couple of fractions off these SECs, and we find that they're rich in nanoparticles. And they can't really distinguish, though, one size of particle from another in the order the fractions come out. And then common precipitation reagents, another common isolation technique. No, you're really going to get all the particles of some unknown solubility to pellet out of the solution together. And again, you get a mixed population from that. So right off the bat, when we use any of these preparative methods, we may or may not be exclusively looking at exosomes from a multivesicular body or microvesicles budded from the surface. And I think everyone understands now what we have is a collection of extracellular vesicles that we need to be able to analyze to determine which are the important ones we want, or what the difference between the different subpopulations is. And the assumption that they are homogeneous in their nature is one that we're going to show in the rest of this presentation is not really a very safe assumption anymore. So it would be useful if there were known surface markers whereby we could pull down one class of these vesicles or get another one alone. The most common markers that we read about, though, are things like the tetraspanins, and differential expression of distinct protein markers between exosomes and microvesicles isn't really noted in their comparisons. What we find often is that people call out the size. I've taken a table here and called out, we'll say the exosomes are 30 to 100. It's kind of arbitrary that they're cut off at 100, but they're small in the intraluminal body. And then microvesicles from the surface should be bigger, or we think they should be bigger. And that, again, it doesn't help us separate them that they're bigger. They need to be different density. And the 30 to 100 versus 100 to 1,000 is all going to come out in the same fraction in SEC. And what's different here is that their origin and mechanism of formation is supposed to be different, but then the only physical characteristic we've described is a potential size difference. And outside of that, they're really all coming out together. So what we have is, in both plasma and in cell culture and in mixtures of the two, we have a wide mix of surface budding and endosomal production being mixed together. So that most of the data that I'm going to present in this talk centers around the technology that we commercialize, the XoView. And I'm going to briefly visit the workflow here, because again, this isn't about it, but for those who are unfamiliar, what we do with XoView is, instead of stepping from the sample immediately to a particle enrichment step, where we do the UC or the SEC or the precipitation, and then taking the product of that and separating our marker realization by either, ELISA, flow, Western blots, immunoprecipitation, and then trying to determine the biophysical nature of the particles we've captured through ISON or Spectrodyne or any type of generic particle counter, which absolutely have their use. We're going to show they're correlated with it. It's hard then to take a look at the particles, take a look at the band in the gel, and not want to make an assumption like, well, if my CD9's distributed all over those particles, that'd be great. And now I know how many CD9 positives I have. What we bring with the XoView workflows, we can analyze by pulling them down, the vesicles directly. We'll size them without a label, and then we can actually co-localize other markers on them, making what very much lets us interpret, like a flow cytometry data set, the subpopulations within the greater bulk. And we're going to show some really nice examples where these subpopulations are real and trackable on the different capture spots. So sample being culture media, plasma serum, urine, saliva, CSF, not really important. What we'll look at mostly today is plasma-derived or cell culture media for our examples. But these hold true regardless of whether you're looking or what biofluid you look in and what type of cells you have. The only thing that changes with the cells is their tetraspanin fingerprint and what extra biomarkers they tend to package on their EVs. So how does this SP-Iris, that's the underlying sizing technology here. So for those of us who are particle sizers, it's single particle interferometric reflectance imaging sensor. And what this is the chip structure. So we have a chip and we incubate the sample on the chip. And when the EVs bind to the specific spots on the chip, we're able to sense that they've bound at the nanoscale below the diffraction limit by virtue of their interference, and we utilize interferometry over the narrow space above the chip to identify the locations and sizes of particles that have bound to whatever functionalizations in that particular spot. And we're able to take these chips and functionalize them for more than one marker at a time. So when we're looking at these pull downs, we're going to actually be looking at the simultaneous from the same sample, probing different sets of particles. What do the CD81 capture, CD63 capture, CD9 capture look like independently? This only senses the size. So this leaves us somewhere in between a particle measurement and a measurement of the protein. We know the protein's there because we've captured them. We can sense their size, but we'd still like to know what other markers these captured particles entrain on their surface. And to do that, we bring in the fluorescent detection. So this fluorescent detection, which I'm going to utilize a lot in the following few slides, lets us reveal that the particles captured on a particular marker on the left, and this happens to be CD9, they can be sized in the left image by the interferometry, and we can show their size distribution. We see it's modal at 65. We're going to see that's a continuing trend throughout this technique. And then over top of that, as a sandwich ELISA style, we can bring in up to 3 colored antibodies to stain the tops. And then this lets us create phenotypes for each of the particles that help us start defining these subpopulations among this very heterogeneous display. So what that looks like is SP iris image on the left, interferometry, calling out a size distribution based on particles stuck to that CD81 CD9 functionalized spot. And then over top of that, 3-color sequential imaging that lets us co-localize the markers into the individual EVs. So what this brings about is a real union of scatter and fluorescence. And we'll touch on this a few times, but many of us have probably run many samples worth of NTA or DLS or Q-nano, and you get that particle feel, but it's often very unconvincing to try and bring in a label into those settings and measure any of those particles as being positive for a marker of interest, even when it comes to the tetraspanin. So we won't directly compare today, but as in past experience, the FNTA method is intuitive, but hardly ever reveals data. So the piece of data I'll start you off with here for the XoView platform is a simple bead experiment. When we put these beads in the system stuck to a chip, they're fluorescent in all 3 colors, and we can detect their size. So upper left, size distribution, Gaussian about 115, looks like a bead. We don't really know if it's right though. Anyone who sized particles, you can get Gaussian about some number and really fail to capture what the actual size distribution is for all number of different reasons. So when we want to question here, okay, how accurate is this distribution? Or should I think that the left side of it is actually relatively smaller to the right? What we can do with these is plot the fluorescence intensities in each of the color channels across the size distribution. So in the plot below the size distribution, we've shown the fluorescence intensity of each of the color channels. And what do we find? An actual second order fit across the distribution, telling us that in fact, the resolution for this sizing technique is certainly under the size of the bins we plotted it in, so less than 5 nanometers. So strong resolution, accurate sizing. We can validate beads with any number of techniques. And then perhaps the most important part for what we're going to talk about here, when we run this control and we look in the plot on the center of the right at the bottom, for the colocalization aspects of this sample, when we look at these beads, we only have one phenotype, and it's the phenotype that all of the beads are fluorescent for all 3 colors. And we're going to find that that phenotype, when we stain EVs or we look at EVs, is present, but quite rare often, and that our double staining and triple staining is a fraction of the different subpopulations. But it's important here to start at a control where the answer is all of the events that I've detected that have a size, and also the phenotype of all of those events is triple positive in the color channels. So that being said, we'll move forward to something more interesting, and that's that all of our results indicate that tetraspanins have heterogeneous expression on EVs, at the single EV level. The chip itself you handle, what I've shown here is just to refresh you on, the capture side is anti-tetraspanins, and we will use the plasma chip. So we'll see plenty of anti-CD41A, and we're going to utilize that to draw some conclusions about the populations we're seeing. And then we can expand these to include more markers and to include controls. They're arrayed on the chip. Chips incubated overnight with the EV-containing sample, and the EVs that display each of the positive markers will be immobilized on that spot. We use the CD41A to assess platelet depletion, platelet-derived EVs. Going to be very important as we go forward here. 3-color sequential imaging, like I mentioned, has given single binding event sensitivity. So this is going to reveal all the small EVs and all the different combinations of tetraspanins that these particles display. And we're going to find out a lot more about that. After EV capture in the cocktail incubation, all you do is dump it in the reader, and we're going to read out this 3-color fluorescence and size distributions. And the colors in this presentation in general are blue staining is anti-CD9 AF488. Green staining is anti-CD81 AF555. And red staining is anti-CD63 AF647. And for these in the kit here, when we look at their staining, we validated all of these with knockout controls against each of the tetraspanins individually. But what I'm about to present is going to show you how colocalization of common exosome quoted tetraspanin markers on multiple EV subsets. That is that although we'll show CD63 as commonly displayed on endosomal derived EVs, we can have it displayed on a multitude of EVs and vice versa. And we're going to find that out through this single EV phenotyping. So microvesicles versus exosomes, endosomal, surface budded. What we'll claim here is that the pathways to surface budding and endosomal formation depend mostly on the available cargo molecules at the site of formation, and that indeed, think back to the table. What we're going to show here is that both of these pathways give rise to vesicles of similar size. So surface and endosomal pathways share mechanisms. The work I'm presenting here is published on bioRxiv by Forjor, George Daubel, our CSO, and Stephen Gould. And what they've utilized in this study is modifications to CD9 and CD63 to study the budding characteristics of HEK293 T cells and NIH3T3 fibroblasts, or mouse cells. We'll focus on the HEK293T here since the result's about the same in both settings. And they're going to utilize modifications which either drive CD63 to localize in endosomes or localize at the plasma membrane, and then look at the budding efficiency of those two strategies. So the first column of images here shows native display at the cellular level of these common EV proteins. And we can see in the top image, CD63 clearly localized in the intracellular compartments. It's not heavily displayed at the surface, as compared to CD9 and 81, which are inside the cell. They're in the endosomal compartment, but they're strongly displayed at the cell boundaries. And then right next to that, we show how converting to CD63 with the Y235A modification, which directs the CD63 molecule to go to the plasma membrane as opposed to the endosome, compared to the wild type at the cells. And then we look at the relative budding. So the cell lysate content versus the intensity of the EV containing both microvesicles and exosomes content. And we find that when the CD63 is directed to go to the plasma membrane, its relative budding, or the amount of CD63 we find outside the cell in the EV fraction, covering both microvesicles and exosomes, increases about 5 to 7-fold. And we can do this in reverse with CD9 and find the same result when they show that the CD9 wild type is on the cell surface, top image on the right, and when the motif directing it to the endosome as opposed to the plasma membranes employed, it quickly localizes in the endosome, and the budding efficiency goes the opposite direction of CD63, which when directed to the surface went up. The budding efficiency of CD9 goes down when we direct it to the endosome, without overarchingly affecting the formation of these particles. So at that point, we've now got an opportunity. If we take the wild type CD63 positive EVs, and we believe that the microvesicles, or those that are budded from the surface, should be bigger, we should be able to use these two cases here to show that wild type CD9 is bigger than CD9YEVM, and that CD63Y235A is bigger than the wild type CD63 particles. So what do we get? We actually find they're virtually identical. We see the same distribution with a mode under 55 nanometers for both the wild type endosomal directed on top and the membrane directed on the bottom. What we do find with the membrane direction when we think, okay, does the CD63 actually influence the particle? Does it make it small because it's a CD63 positive vesicle now? And maybe it thinks it's something like an exosome and it's not a big microvesicle. We can use the CD9 wild type to discern that, right? We know much of the CD9 is coming from the surface, so we should be able to look at the wild type CD9 EVs and see that. We'll note here also that when we direct the CD63 to the surface in the presence of wild type CD9, we see huge increases from about 20% up to about 80% in the colocalization of CD9 and CD63 on the same particle. So this is very suggestive that these pathways are in fact distinct. We have many CD63 onlys that come from the endosomal pathway, but at the same time, when we direct the CD63 to the membrane, we get a new class of particles that entrains much more 63 and 9 on the same particle. So we go look at CD9 wild type to check if they're bigger. And in fact, they're not. The wild type CD9 captured EVs don't really show any size difference to those captured on CD63, even though they have local localization with CD63 and would seem to be budded majorly from the plasma membrane. When we look at the endosomal changes, the endosomal direction, we decrease the relative budding, but we don't influence the size distribution. We get still small, similarly sized vesicles. So all things being considered, are they bigger or smaller, or what is their exact size may not be discernible. But the fact that these are the same size is almost certain based on the intensities they give us back from the interferometry and the size distributions that can be perceived when they're bound based on the wild type display of the normal tetraspanin or the display of this endosomally or membrane directed modification. And we find the budding changes appropriately, that plasma membrane budded vesicles dominate in either case, and the endosomal membrane vesicles are the lower fraction, and that their size is not really influenced by the tetraspanin content. The incorporation of the tetraspanins is mainly driven by their local availability, depending on where they've been directed to. So the conclusions from this study, and this is available on bioRxiv, are the exosome-like tetraspanin-rich particles can bud from the surface of the plasma membrane and also form in endosome compartments. The relative budding frequency indicates which tetraspanins are inherently endosome-directed. Naturally, we see that CD63 here. And that modulation of the subcellular localization motifs can change the budding frequency, indicating that location within the cell and not necessarily its function in the formation step is the major driver of the different phenotypes that we see when we do this experiment on naturally occurring EVs. Using SP-Iris and the fluorescent imaging, it's shown that vesicles which bud from the plasma membrane have virtually identical size to those from endosomal compartments, even though they have completely different tetraspanin compositions, and that manipulating the tetraspanin composition of any of those subsets by redirecting the tetraspanins to a new subcellular location doesn't affect their size distribution. We can take this another step further and look at the colocalization of tetraspanins in human plasma and cancer cell-derived EVs. Here's something familiar. Some beautiful NTA videos. And what we have here side by side is two different plasma samples. And I think a lot of us have probably done this experiment before, gathered data like this. We're looking at NTA to tell us about the total nanoparticles present. And when this blood product derived sample has EVs in it, they're often just a subset of a complex mixture of other nanoparticles. So I've borrowed this lovely graphic, which I think we've seen in many different forms from different groups. I took Simonson's graphic here, and you can see immediately there's multiple size overlap. So protein aggregates can certainly cover the whole range of sizes on the x-axis, but of particular interest, VLDL, chylomicron, some LDLs completely overlap the exosome and microvesicle content in terms of size distribution. You can see they sit right on top of the same distribution. And so when we do an experiment like this, we get a beautiful result. This says that there's 3 × 10⁹ particles per mil. It says they're 88 nanometers at the mode, and that D10 to D90 is 63 to 183. These look a lot like what we would expect a vesicle fraction to look like. And then right next to it, we have another plasma, and it's very similar, but it has less material in it. It's got it reports 6 × 10⁸, so about 5 times less material in terms of total nanoparticles. What do we really know about the particles from there? This is where assumptions come in. It's easy when you look at some of these types of data to say, okay, I can see all my nanoparticles there, and I would like to imagine I could label them all for a marker of interest. And I think it's often difficult to do. Here it's intuitive that you could put the filter in the way and see the particles, but it's not usually successful for sensitivity reasons, or perhaps because in some cases you can be looking at many nanoparticles that aren't in fact EVs if you don't have a positive tag on them when you have a complex mixture. So keep that fact in mind And we're going to explore that a bit more as we go on here. So when we run these two plasmas, which we just looked at by NTA, they're very different. They had five times the smaller particles in plasma 2, more particles. Note smaller by NTA. They went down to 88 in the additional particles. What we find when we put these through our assay for XoView, we get these label-free charts on the left. So this would be size distribution. Is there binding over the IgG here? And what we have on these chips is a CD41A spot. And CD41A is an integrin that is intrinsic to platelet-derived microparticles. And when we look at the response of that spot, we actually find that the tetraspanin spots themselves, even though these samples look quite different in total particles, the tetraspanin spots respond very similarly. We have similar counts, similar phenotypes. We've got a nice red-only phenotype on the CD63. We've got a lot of CD63, CD9 colocalization naturally. But in the top row, note this lack of CD41A response, okay? We've got almost no platelet-derived microparticles present at a detectable level, even though we have thousands and thousands of EVs present. When we put sample 2 on, bottom line, we see a big response on the 41A spot. We develop this. Particles, secondarily, we label in the blue bars with CD9, and we label with CD63 in the red. So what does this actually show us right here? This shows us that platelet-derived microparticles display both CD63 and CD9 on their surface. And we see that change as a percentage of the total EVs captured here in the table. When we look at the fold change between the two, we see that CD63, CD9, and CD81 capture within 2-fold of the total number of particles. But we get 13 times the capture on the CD41A spot, indicating that plasma 2, in fact, has many more platelet-derived particles present in it. And I think that's an important part of realizing that the analysis of this sample for CD63 bands would have revealed the same result for both, but the location of the CD63 in plasma 2 is almost equally on CD63 vesicles and CD41A vesicles, and the platelets have contributed a significant fraction of the material in terms of vesicle content to plasma 2 here. We can again look at the sizing to help us understand if these are really the same particles. When we look at the CD9 size distribution with low CD41A, so that'd be the CD9 capture on plasma 1 on the top, we get our distribution down to small size. It's a little chunky. There's not a ton of material there, so statistics could be better. We might be expecting that the particles get bigger. If we look at the CD49 or the CD41A size distribution, we might think we're going to look over there and see big particles. But we in fact find that these are all sized at around the same area, under 100 nanometers, modal at 55, regardless of whether we look at the CD9 size distribution under high CD41A, so that's those captured by CD9, or the CD41A size distribution under high CD41A. So we've got thousands of particles down there. Are they bigger? There are big ones. There's hundreds to 200s, but the vast majority of these particles that bind these spots are sub 100 nanometer. And I think these results as a whole here highlight the need to screen for platelet-derived particles in plasma and serum samples, because in the case of plasma 2 here, the platelet-derived particles are contributing at least 50% of the vesicular signal. And we can see that shine through to us in an interesting way on the CD63 spot, actually, by using a colocalization analysis. So the pie charts here show how the phenotypes of those particles captured on the CD63 spot are broken up. And we see that in plasma 1, which has low platelet-derived microparticle expression, the vast majority of our CD63 captured particles only counterstain for CD63. And we get about a quarter of them that co-stain CD9, which we can clearly see happens. We can see the CD9 spot and trains plenty of CD63. But looking at the pie on the right for plasma 2, we see a big expansion of the pink slice, and that's the CD63, CD9 colocalized slice. And then we see a big expansion of the CD9 stainers in addition, and that goes right in hand with the idea that there's a big class of CD41A platelet-derived particles that have showed up, and they're positive for both CD9 and CD63, and thus they start to show up in your analysis on those capture spots. And you can see it really clearly here with the enhanced colocalization of CD9 onto the CD63 spot in the presence of many CD41A derived particles. So that's all fine and good. You know, that's interesting. We've got the same size particles coming from everywhere. We can entrain multiple of these common exosome markers on them. Even platelets can butt off particles that are small and display 63, 9, and 41A. So what about markers that are a little more interesting? You know, a cell surface marker like HER2, common cancer cell marker, a lot of research on market drugs targeting it. This is interesting. We take a cancer cell line here, SKBR3, this is typical work. We grow the cells in a dish, we isolate them, and chip 1 on the left, I've stained the tetraspanins. There's nothing surprising about that except the lack of CD41A. When we run these, anytime you run a cell line that isn't platelets, you'll get no response on the CD41A pad. So that really enhances our view that, is that differential fraction we capture in the plasma actually driven by the platelet particles? Absolutely. So here's our tetrastaining. And then next to that, you'll see the blue bars are almost exactly the same. It's the same sample. So CD9 staining stays the same. We've left the green dye out, and we've actually incorporated anti-HER2 here. And what do we find? Shockingly enough, the HER2 strongly colocalizes with the surface bleb particles containing CD9. HER2, known surface marker, needs to be a homodimer on the surface for activation. And sure enough, here we find it strongly colocalizes in the surface budded, largely CD9 particles. And as a percent of the total capture, it represents almost a quarter of our CD9 particles on that spot. So this is a strongly expressed marker in these EVs derived from these cancer cells. And we can see it at the individual EV level here. We see the overlay of the red and green. We can see how strong the red is. When we examine the display of the HER2 on the CD9 vesicles in detail, we find that the display and entrainment of the HER2 correlates with the vesicle size, which calls back to its inclusion of biomarkers on the surface release being driven by their local availability. So however many are available for the size, the more surface area you bud off, the more HER2 you should entrain. That's exactly the trend we get here. So we looked at some NTA from the plasma, and to be fair, we know that plasma NTA is tough. We showed a chart that showed there can be many contaminating particles, but maybe in our cell culture fluid, there's not. Maybe when we do cell culture media, we can be a little more firm that when we NTA the sample, those particles are actually probably are EVs left from the cell because there's not so many contaminating particles. And what I've shown here now is this, instead of plasma, cell culture media derived EVs done by NTA, red line, and then I've plotted our size distribution over the top. And there's some overlap. You know, these are following the same range. You can see we detect a lot of small particles on the XoView platform compared to the NTA kind of Gaussian about 100 distribution. And that's on the left. So as a number weighting, we show a very different distribution. Then if we try and capture the concentration, so if we try and say, when I measure by NTA, this is how many particles I think I should bind, and we look at our size distribution counts under that, we end up saying, okay, well, I've got something somewhat similar, but I don't have very many particles total that I might expect. And this makes you think that there's a possibility, maybe these ones I sense in NTA are contaminants, but maybe they're not. Maybe it's just driven by the hydrodynamic versus the dry sizing. When we do the XoView method, they're dry on the chip. So maybe it's a hydrodynamic versus dry sizing thing, but there's one more aspect we haven't really discussed, and that's that these tetraspanin signals, when we use the fluorescence mode, are really prevalent in what we think of as small EVs. And we know they're small because here we see our size distribution in the upper left, and I'm showing you a single color channel image wherein the pink circles are those that are size. They're captured for their size. They're measured in the size distribution by the label free. And then the blue circles are those CD81 positive signals and very well can co-localize with secondary tetraspanin signals that are not, they're just not in a sizing regime that we can measure. And then below that, I show the image, the interferometry image that drives that. And I've circled those areas where we can clearly see 5 or 6 events that just don't have a corresponding size distribution. So if they were bigger, if they were in fact aggregates or they were on the big side, we would clearly see them in the interferometry regardless. So we get no signal in the interferometric image. So at that point, we know that these have to be small. And we see these both in the plasma situation, the cell line situation, and in basically every isolation method. There's a whole contingent of these things that are small, but we're going somewhere with this. They're undersized. We know that hydrodynamic versus dry may not be the easiest correlation to make, but what we can do when we have these, instead of plotting just our size particles, we can now incorporate these fluorescent ones into the sizing. We can talk about them as EVs. And what I've done here with the HER2 staining is showed that this isn't just a tetraspanin cluster. It isn't that we have a homodimer of CD9 that we stain one side of and then stain the other. Because when we stain for HER2, completely secondary marker, totally extraneous to the tetraspanins, very focused on this cell line's EVs. In fact, with that, we still only have about the same 35% percent of the particles that fall in our size range. And that's the red spray chart on the left there. We see clearly this HER2 intensity. We see clearly that it's correlatable with size, but in fact, we still have many HER2 positive EVs that fall below the sizing limit we sense in this distribution. And where we're going to close this up at is that when we include those, so if we think of our total as the total unique fluorescent and sized events on a capture spot or within the context of the experiment, and we add up those populations, for homogeneous samples like cell culture media, when we include the small particles, we can often reconcile the count, but show a shift in the size distribution. So we find they're small. I've plotted them as artificially small here. But if we interpret them as a total, and we ask how many does that imply in the solution, now when we take our SP iris and fluorescence combination and we convert back and we make an estimate, we would call out here that there's 4.1 times 10 to the 7th per mil. And in fact, our NTA measurement says it's around 5 times 10 to the 7th per mil. So in this homogeneous cell culture driven environment, we can think of them as part of a total, but in the plasma, even with isolation, there still remain many particles where this type of analysis will leave you thinking that only a fraction of the plasma nanoparticles are in fact EVs that I can measure, whereas in cell culture media, many of them contain the tetraspanins on the EVs just like we imagined. So the takeaways from this section, which is quick hitting and kind of information dense, are that plasma EVs, anytime that you have plasma, urine, any biofluid, there are various populations mixed together that are from functionally various origins and different sources. Common exosome markers can be detected on platelet-derived microparticles, including CD63, and these are likely often co-isolated with exosomes when you have these type of fluids. And as we visited in the previous section, the surface release particles tend to entrain surface displayed molecules, and we find the colocalization of HER2, surface displayed cell marker, and CD9 is stronger than the other tetraspanins for capturing HER2 positive EVs. So maybe not intuitive off the bat, but when we know more about the mechanistic nature of the CD9 released particles, it makes perfect sense that that's where we would see the strong correlation with the HER2 display. And although the total particle results often correlate with the response in the sense that we could sense there was more material in the plasma 2 versus plasma 1, revealing the relative quantities and contributions of the different subtypes to capture what those phenotypes are can only really be revealed when you can probe the EVs at the single EV level for what their biomarker status is. And what we found was plasma 2 had many platelet-derived microparticles in it that actually contribute signal to the other normal tetraspanin capture spots without revealing that they're platelet-derived if you don't check for CD41A. And lastly here, I'm going to visit a spike-in experiment where we'll use this colocalization of tetraspanins to actually track cancer-derived EVs spiked into plasma and talk about when these two different capture strategies can be advantageous relative to one another. So finding the cancer subpopulation among heterogeneous plasma EVs. So now that we know that all of these different EVs can display the tetraspanins, how can we use that to our advantage if we have disease EVs and we want to look at disease EVs? Can we pull down on, is pulling down on CD63 a really, yeah, there we go. Is pulling down on CD63 the best way to capture the cancer EVs, or is it better served with a secondary biomarker? And we'll pick this apart a little bit. So there's two detection approaches we're going to analyze. We're going to spike in ASPC1 derived EVs into whole plasma. And ASPC1 CD63 positives display very strongly T-span8 on those EVs, and T-span8 is not detectable in the whole plasma. So we're going to spike this into healthy donor plasma, and we're going to use two approaches. There's the one where we capture on CD63, like we have been with CD9, and then looking at, in this case, ASPC1, or it could be, it's akin to the HER2 experiment. But what we'll do here is we'll also capture on T-span8, and there we're going to remove the predication that these EVs need a tetraspanin in order to come down on the spot. There we'll just capture on T-span8, maybe probe for the tetras, probe for T-span8, and see what our counts are like. So two different detection schemes, and how do those help you when you dilute this through? So starting on the left, high concentration of cancer-derived EVs. So this is the full concentration. The image on the bottom is the T-span8 capture. So that's captured what you would expect are only the spiked in EVs. And the top is capturing both the spiked in EVs and the native EVs based on CD63 at a constant amount of plasma. So we see on the left-hand side, it's very bright red, and we count 6,000 T-span8 spots. So the bright redness there is actually the addition of the cancer. We can see that the ASPC1 derived EVs are adding to the CD63 signal in a very relevant way. So if that's the case, you can obviously detect it with CD63. Or secondarily, we can count all this T-span8, and that's a trivial situation, right? We've mixed them roughly 50-50. We get a big signal enhancement, but maybe not to biologically relevant. We're really looking to go more to the low end and see which of these strategies at the low end gives us better sensitivity to the small subpopulation of T-span8 particles. So you can see as we dilute to 27x and 730x relative to the plasma, our CD63 signal starts to look the same in the red channels. That's the baseline of the plasma. That's the CD63 positives in the plasma. And among those, we can count some T-span8 positives down to about 0.5% of the total events on the spot. When we're looking at CD63 capture and trying to counterstain a marker, 0.5%. And below that, we need the 2 to 3-fold signal enhancement that detecting based on T-span8 gives you in order to see that we have these populations in. But in turn, that gives us 2 to 3-fold more sensitivity when we use a custom capture spot. So when you have rare events, it can often be advantageous to have them. And we might say, well, what ratio do they need to be at? Because if I'm going to use the CD63 spot and they're 1 in a million, I'm going to need a million EVs stuck to the spot. It's going to coat and it might not sample enough EVs in that case. So another situation where using a custom capture spot like the T-span8 here affords you greater sensitivity as a relative part of the total down into the 1 in 100,000, 1 in a million range by concentrating the sample, knowing that it would saturate a tetraspanin spot, but you've got an appropriate number of disease positive EVs to come down on the known marker for their capture. And really nice, easy experiment here that really captures the difference between sensing EVs in the plasma and sensing EVs derived from the cancer cell based on their specific marker, T-span8, a different tetraspanin here. So this is, I'll recap it here. I'm at almost an hour on my timer, so I think I'm getting pretty close. Widely seen as classical markers of EVs, we've all been researching, talking about these, measuring them for years. It's very interesting to me now to look at the display of the tetraspanin family of CD81, 9, 63, and how it covers multiple subsets of EVs that are obviously released from various origins. We can wrap that up with just the platelet-derived microparticles, and that they can be co-expressed on the same particles released by different cells. So at a baseline, looking at CD63 or CD9 display independently can't isolate a single population of EVs. The availability of proteins at vesicle formation sites drives their incorporation regardless of the pathway. So for the EV engineer, for the vesicle engineer, this is important. More expression on the surface will lead to more entrainment in the CD9 positive particles and the surface budded particles. The localization of different tetraspanins within the cell and their cargoes drives their colocalization with additional markers of interest. And that tracking specific subpopulations within normal plasma and sera is possible when you have the tetraspanin pull down in a specific marker, and even better when you know a marker of interest that corresponds to your disease state or EVs of interest, and you can capture them specifically. So I thank everyone for watching. I'm happy to sit and discuss, answer some questions. If you'd like to learn more about the platform or any of this work, we have a comprehensive list of publications on our website, and you can visit nanoviewbio.com. And with that, just we'll open up the discussion.
Thank you very much, Clayton. Wonderful talk and very informative as well. And, we're very excited with this new technology. So we have first question is from Phil. Wondering if Phil is there. Okay, I'll just read it out. Have you found immunoglobulin light chains on the surface of some exosomes or EVs?
I can't comment because I haven't stained for them in the way that was described here. So couldn't say.
Okay, yeah. That's right. Reginald.
Hey, Clayton, very nice. So I might have talked with you about this before, but I was just curious looking at your work again. Can you use this, say, ratio of, I'm going to use an example, CD63, CD9, since people think that that has, prognostic significance in some cancers. Is there such a thing as kind of a EV ratio footprint that you would say, like, say, just a simple cell line might have? And could that be altered by the presence of another cell or even heterogeneity of, cell line types? And so I'm just wondering, how sensitive do you think the ratio of any of these tetraspanins could be for identification of certain populations of cells?
In my experience, very, very possible to use where you can almost look at a cell line as having a fingerprint, and that BT474s versus MCF7s versus ASPC1s, if you had seen them for their tetraspanin colocalization patterns, you would be able to tell after the fact which was which, and that growing them together would give you a mixture where you could hope to approximate based on the cell number what the contribution of each one was. So that is certainly a reasonable question, and one that definitely gives you data that you can explore. As far as the CD9, CD63 colocalization aspects, you'll be able to sense when there's mixtures of cells or changes to that population induced by some other stimulus to the cells.
I'll ask you here to kind of hand wave if you want. Do you think that it's possible for a cell to learn or mimic the tetraspanin pattern or release characteristics of another cell line? Is that something that's intrinsic or extrinsically controlled?
As I do not have enough data to hand-wavily answer that. They can change, right? So external stimuli will certainly change the phenotypes, but do they learn and remember them? I do not know enough to say.
Thank you.
Actually, following to that, Clayton, do you know or notice some sort of differences in terms of the profile from different cell type, like epithelia versus immune cells, whether there's sort of like different colocalization pattern and so on?
There are certainly patterns between them. You know, endothelial versus cancer cells versus immune cells, they do all have different patterns. And when you culture them distinctly, they make a fingerprint of what their EV touch. Some are very high in CD81 and emit tons of CD81 only particles. Others display almost no CD81. So certainly they all have their own fingerprint for what their natural tetraspanin display looks like.
Okay. Nicole, please go ahead.
Hi, yeah, I'm here. My question was just about like the picture of the display of the data for the colocalization, the fluorescence colocalization. Like I noticed there was some kind of like black box kind of on the image there, and was just wondering what that was.
Yeah, easy one. They're just fiducial marks for alignment. So the chip is 1 centimeter by 1 centimeter, and when we're looking at those colored images like this behind me, this is 200 micron from edge to edge on the spot, and those L's are just alignment tools for the images.
Perfect. I did have one other question that I didn't get to put in the chat. Sorry. So I was kind of surprised at the mode size for the tetraspanin EVs, tetraspanin positive EVs on the chip. They were about like 50 to 60 or so. And so I'm wondering, again, knowing that maybe like microvesicles of larger size can also display these tetraspanins, were the samples size filtered, or like does the chip itself and how it works kind of preclude binding to larger EVs?
It doesn't preclude binding. If you thought there were far, far fewer, you could be in a regime certainly where you preferentially bind the small ones because they're so much more numerous. There's no bias to it though. If they were practically all 150 nanometers, I think that's kind of the result that George and Steve Gould and Forgeorge showed, is that if you, even in the wild type, if you thought the surface budded ones were bigger, that's definitely the result you would get. And so even if we don't know if that's the exact right physical size for comparing to NTA, we know with certainty that it's exactly the same as the ones from the endosome, because they give exactly the same contrast when we look at them under the SPI iris. So their size, is it 50 or is it 70 by NTA, or is it 80 and it's 55 in ours? It's the same in both is really the takeaway. And it is surprising. I think for a long time we've thought the surface budded particles are bigger, the microvesicles are bigger. And the data displayed here really kind of suggests that the majority of these things are the same size and probably limited by lipid curvature questions and protein entrainment questions, more so than the idea that, oh, they're blebbing, so they're bigger. If they're controlled by the lipid composition and the protein entrainment, they end up looking very much the same size. The question is that the exact size? Is it 65? The relationship between hydrodynamic diameter and the diameter in SPI iris where the chip is dry is probably if they come out smaller in ours.
Yep. Okay. Thank you.
How about the sensitivity, Clayton? Can you sort of differentiate like vesicles that are about 20 or 30 nanometer in diameter, like the difference?
The difference, absolutely. We can see with the contrast method on the slide that showed the beads that the sizing method is certainly accurate under 5 nanometers as it pertains to contrast. So 20, 30 nanometers is trivial, and we have tons of mixed beads. There's a good paper from Ken too that We could ask whether that's important, certainly whether discrimination, but the case of the SB-IRIS system and the methodology for particle size in mixed groups, R3 relationship, and everyone's well on scale. There's no occlusion. Having a 200 next to a 50, there's no occlusion of the signal of the 50 by having the 200 in close proximity. So the result we get just says that the prevalent population is the sub-100 fraction.
All right. There's one last question from Belcher. Could you please unmute yourself?
I know who it is. Was it John Belcher from University of Minnesota has asked the question.
Right. Yeah,
have you looked at EVs derived in endothelium and plasma? Short answer, no, we look mainly, do we know, we're looking at the tetraspanins, there could definitely be some endothelium derived in there. Without the additional endothelial markers, I think it's hard to say. I think this definitely shows it's hard to say for sure. It would be intuitive that many plasma EVs are hematopoietic, but when we see the tetraspanins are co-displayed on many different sources, and we know they're on blood cells, immune cells, and platelets at a basic level, there's no reason to think the endothelial cells are not using them also. What we need there is endothelial markers to sort out which part of that population is actually coming from endothelium.
Hi, Clayton. Thank you very much. We have worked with George quite a bit. So two major questions. Everything looks wonderful the way you presented. From the cancer point of view, from the way we have tried to identify cancer-specific EVs, we have used a mouse model, knock down the cancer-specific integrin in this particular case, and then have gone and look for the presence of EVs that had this particular marker or not in the plasma. So that is possible. You know, your spike, your attempt to spike is also confirmed by our mouse model. So the only thing you might want to look back at our paper is to see if there is what percentage of vesicle really come out that will allow you to be sensitive in a cancer patients in vivo, in the humans, right? That's what
I'll look closer at that. Because you could see it in the mouse when you use the mouse plasma.
Yes, yeah, because we did knockdown, so it was cancer specific, epithelial specific. Carolina worked on cancer, on prostate cancer, so it's something that it's very unique, a Trump model where you can knock down the integrin specific in the epithelium. But then I have to ask you a question back on the paper with Steve Gold and George. I was, while you were speaking, I was looking for it. I haven't downloaded yet, but what was not clear to me is that the CD63 is only in endosomes specific, exosome derived from the endosomal compartment. Is that what we're saying? Because you guys forced expression on the plasma membrane.
And in the wild type, it's strongly localized to the endosomes only. So there is certainly a level that you see in the plasma membrane, but the change and the co-localization with the CD9 after modification suggests that more than 80% of the intrinsic CD63 is in endosomal derived vesicles.
Endosome, because we see a CD63 on the surface of the cells, you know, we don't really even have to go, so it's there. So that's a little bit of a
detectable, certainly. It makes up a fraction. It changes significantly when you direct it to the surface. And it becomes a new phenotype that says, oh, now I see 5-6 times the CD63 I saw before. But certainly in the wild type, there is a very detectable group of CD9, CD63 positives.
Okay, perfect. We all agree on that. Very good. Thank you. Dan.
Yeah, no, it's not exclusive. It's not exclusive. It's just a major fraction.
Yeah, but the paper is still not in press, or how is it?
It's available online on bioRxiv right now. George would have to give more of a status update than that, but it's on bioRxiv in its entirety.
Okay, very good.
The figures I showed are in the paper.
They're from there. Very good. Thank you very much. Thank you, Clayton. Very nice. We must have met when you were at NTA. Did you come at Jefferson?
No, I covered the same area. So always West Coast. Always West Coast. I like it warm, sunny.
Yeah, better. Okay, perfect then. Thank you very much. Thank you, Carolina. Great job here.
Yeah, thank you very much, Lucia.
Thank you.
All right, if there's no more additional questions, I think we'll thank Clayton for exciting data that you presented here, and also raising, you know, more questions in terms of the scientifically, what is this CD63, CD9, CD81, and their function in the cancer and other diseases. And actually, I was just reminded now, have you actually looked into urine versus plasma and whether the colocalization changed and so on? Maybe
I can tell you it does change. I didn't present any data. It is different. Like urine versus plasma, it is different, but you still see those same characteristics. There are still CD9s on CD63s and CD81 CD9. Urine tends to have a lot more CD81 than the others.
Oh, okay. Well, that's
Or
another exciting thing.
Plasma, it tends to be high in urine, but yeah, maybe I'll make another talk out of that, and we can do different biofluid tetraspanins.
That's true. Yeah, that will be exciting. Okay. So yeah, again, thanks so much, Clayton.